Finale. So I'm delighted that we have Dr. Joan Fujimura to be here with us on this occasion. This is also my last opportunity to thank everyone who helped make this possible. So I'd like to, it's a very, very long list of people that I truly need to thank. But I would like to start with thanking Julia Hutt, my student assistant, who is the author of all those beautiful posters. So thank you, Julia. The posters have been bringing you here, but Julia also helped me all along with all the less glamorous details of this organizational affair. So many thanks, Julie. I'd like to thank many of my colleagues here at King's, and I have to say I'm very fortunate to be at the institution that, even in the times of so-called fiscal constraint, does know its priorities and gets its priorities right, and through its weight and its money behind this event, although obviously this would not have been possible with numerous sponsors and benefactors who also helped out. So I'm really grateful to my colleagues in the upper level programs at King's, Contemporary Studies, Early Modern Studies, History of Science and Philosophy, and School of Journalism. They all help in different capacities and to the best of their abilities. I'd like to thank President Barker, King's Center of Interdisciplinary Research, as well as Brian Fleming. These are the groups of individuals that came to the rescue of the lecture series when we kept running out of money. So again, my gratitude goes to them. I also owe a lot to my colleagues from the Dalhousie University in many departments. I made a lot of great friends along the way, so just one of the benefits of being an organizer. My colleagues from truly a number of departments, but on many occasions, history department, philosophy, theater, gender and women's studies, and many others, helping not only financially, but by coming here regularly, bringing the students. I am even aware one of my colleagues even checked attendance at the lectures, so that was very helpful. Special thanks and a special occasion of this lecture go to Situating Science, no longer the poster, but I'm sure it's etched itself in your memory. So many thanks to Situating Science Knowledge Cluster, which is a national strategic knowledge cluster run out of King's College. Thanks also to Evolution Studies Group of Dalhousie University that so generously sponsored this lecture and another lecture in the past. So again, we would not have been able to do that without that generosity. I really hope that the whole lecture series, which as I say has been a long journey, will leave some legacy behind. I hope it will open the door to many more events of that sort, to a conversation, and at least from my point of view, I have learned to think differently. I have learned to look at what's around me in a new way. So I hope this will be a legacy of this lecture series. And again, I thank you for coming. I would now like to welcome Lisa Gannett, who will introduce the speaker. And Dr. Gannett is a professor of philosophy in the Department of Philosophy at St. Mary's University. She's also a member of the Evolution Studies Group. Her research intersects very closely with Dr. Fujimura's in the history and philosophy of biology, and she's also interested in the use of group concepts such as population, race, and ethnicity in genetics and genomics. So I'd like to welcome Lisa Gannett, introducing Professor Joan Fujimura. Thank you.
My computer went to sleep.
I'm very
have the opportunity to introduce Dr. Joan Fujimura to you this evening. Dr. Fujimura is a professor of sociology in the sociology department at University of Wisconsin-Madison. Before this, she held appointments at Harvard and Stanford. At Madison, Dr. Fujimura is affiliated with several other units, including the Holt Center for Science and Technology Studies, Asian American Studies, and Gender and Women Studies. I had the opportunity to be a guest of the Holt Center in November, and I can tell you that this is a place that has managed successfully to pull sociologists, anthropologists, historians, philosophers, and natural scientists into fruitful discussions, as we are trying to do here in Evolutionary Studies Group and Situating Science Cluster. You will appreciate in her talk and during the question period how easily Dr. Fujimura herself manages to cross these disciplinary boundaries. Dr. Fujimura will talk to us this evening about the results of an ethnographic project she began in 2007. Dr. Fujimura and her collaborators spent time in 5 different laboratories, all of which were involved in research on human genetic variation, with the goal of understanding how the concepts of race and ancestry are being taken up and reconfigured in the course of this research. Dr. Fujimura is spending this year at the Russell Sage Foundation in New York, where she is working on a book. Her 1996 book, Crafting Science, A Sociohistory of the Quest for the Genetics of Cancer, won the 1998 Robert K. Merton Prize awarded by the American Sociological Association. This current book on genomics and race, Dr. Fujimura intends for a general audience. Writing such a book is, of course, a challenge, and I know she will welcome all of your questions because this always helps the writing process.
Thank you, Lisa. And thank you to Rotha, thank you, Gordon, and thank you, Ford, or Dr. Doolittle, as I always say. I really appreciate the invitation to come and speak in your series, and I looked at all of the other fonts in the list, and I was very impressed at your speaker series. We, unfortunately, can't afford to do anything like that right now, but we will try. You've set the example. Okay, so I do tend to forget my slides, so just holler slides if you find that I forget. So I'm a sociologist of science, and I do cross boundaries easily, as Lisa said. But there are times when my sociology colleagues want me to stick to sociology. So we all face this issue. But as a sociologist of science, that means that I examine how knowledge is produced. And I especially focus on social and scientific issues, how they come together. My most recent research is focused on how race has recently reappeared in human genetic research in the U.S., and especially in biomedical genetics research. So that sets the context for our research project. Then I'll talk about our research. Basically, our question was when and where is race used or not used in biomedical human genetics research. And then I'll talk about, I'll end with conclusions, but I'm not sure if they're conclusions. I might have to call them discussion points. And so I welcome all discussion on the points that I'll raise at the end. So many sociologists, anthropologists, and biologists have long argued that race categories, as proposed by early 20th century anthropologists, do not constitute biologically or genetically distinct populations. So building on these earlier studies, later scholars viewed race as a social construct and race categories as socio-historical categories that have developed over time in particular locales. So let me note here that this view of race as socially constructed doesn't mean that it's ephemeral. Right? Race is real in its consequences. And so don't take it to me like I'm talking about something very ephemeral. The scholars argue that race categories differ by locale and change over time. Most sociologists have argued that ethnicity and race should be conceptualized not as substances or entities or even groups of individuals, but instead in relational, processual, dynamic, and disaggregated terms. Sociologists of race studied race and ethnicity in terms of the practical constitution of categories as cultural idioms or as cognitive schemas, as discursive frames, as contingent practices, and as political projects. These are all, look at the sociology of race at some point, and you'll see that there's so many different ways to study race and to conceptualize race. So these scholars argued that we should analyze how races are formed, that we should treat race as a mode of analysis, and not as completed categories. They argued that we should examine how race among various publics often, how race, the concept of race among various publics often conflict with official race classifications, like those of the U.S. Census Bureau. I think you know that in the U.S. we collect data by race categories. Other studies discuss the differences in race classification systems between countries. For example, Brazilian race categories are very different than American race categories. Let's see, there are also, other sociologists have studied the differences within classification systems in a particular nation between particular time periods. So one of my colleagues in Madison is looking at race categories in Brazil historically. Let's see, and I should note, for example, that the term race is not used in Germany because of the history of the Holocaust, and it's also not used in France, in part because the French government assumes that all people are French citizens, including the recent immigrants from their former colonies in Africa and elsewhere. So there are many different ways of thinking about race. So in other words, scientists have argued that ethnicity, race, and nationality should be studied as political, social, cultural, and psychological processes. There were still sporadic efforts to argue for biological and specifically inherited differences between races in, for example, intelligence in the second half of the 20th century. But these arguments were put forward primarily by individuals without any scientific evidence and were largely dismissed by scientists. That, of course, doesn't mean that it didn't have a life of its own in the general culture. Now that's something that we deal with. In the last 10 or 15 years, however, race categories have resurfaced in biological and medical research. Let me just see if I'm forgetting those slides. Okay, no, it's still fine. Some researchers have argued that there are biological differences in health, disease incidence, and drug response between different racial and ethnic groups, and that knowledge of these differences would be useful in medical diagnoses and treatment. Esteban-Burchard in particular argued that to ignore race would be a disservice to those who are underserved in the American population, and he was specifically speaking about Latino population. In reaction, critics of this research argued that the search for genetic contributions to racially stratified health outcomes has distracted clinicians and public health officials from more proximate social causes and more effective social interventions to redress these inequities. Other scholars argued that while race as a socio-political construct is appropriate for monitoring health disparities, it's not a good tool in genetic studies, especially of common complex diseases, which is what I'll be talking about today. So what is wrong with using race categories in biomedical genetics research? Indeed, some scholars have argued that race may be useful as a proxy for anything from diet to ancestry. But they go on to say that contemporary biomedical uses are often incorrect, misleading, or may cause more harm than good. Their primary concern is that the conflation of especially U.S. race categories with research categories in human genetic variation and race-targeted medical and pharmaceutical research has the potential to reinstitute and reify biological categories of race. So the debates and concerns reveal a diversity of views about how to interpret recent genomic studies of disease, causation, and treatment that use notions of race, ethnicity, national origin, and ancestry. The scientists who engage in biomedical genetics research have also diverse ideas about this issue, as I will talk about. The debate, they're not all the same. So the debate about the use of race as a research variable is also not limited to the biomedical sciences. Related debates in the disciplines of anthropology and sociology about race and whether or not to use race categories in our research have been heated and have led to the production of official positions on the definition of race by the American Anthropological Association and the American Sociological Association. And the responses of non-academic publics are also mixed. Some studies have shown that new genomic challenges also affect how everyday humans, how we classify ourselves, while others show that individuals have complex resistances and attractions to genomic information about ancestry. I don't know if you watch Oprah here, but Oprah had always thought that her family was Zulu in origin, and she was really distressed to find out that they were not Zulu in origin. But there are issues with that test, so I was going to say she shouldn't believe that, but we're live streaming now, so now I want to talk about our research. So where and when is race used and not used in biomedical human genetic research? So as I said, the use of words like race, ethnicity, tribe, nationality in biomedical genetics research galvanized a host of critiques from bioethicists and social scientists. But it wasn't clear who was actually using racial and ethnic categories in their research, and if they were using them, how they were using the concepts in their research processes. It was clear that they were using it in their publications. And Pamela Sankar and Mildred Cho wrote a very good article calling them on how they used the term in very sloppy ways, and that they really needed to clarify what they were actually talking about. But when we started our project, there was no one, well, one of my former students was working on this, but there was very little research on how race was actually used in the scientific process. So as a sociologist of science, I decided to contribute to the conversation by examining what was happening in genetics and genomics research practices. So in other words, I decided to study who was and who was not using race categories in their science, how they used it, why, and why not. So in my kind of sociology of science, we don't only read published works, we actually go and study the scientists at work. It is an imposition. We do stand there in your laboratories and take space and observe you at work, because it's allowed by So for this study, we decided to focus on, well, I decided to focus on research labs and centers that had the best reputations for their science. Some of my colleagues in the field were studying some of the more problematic laboratories, scientists who did use race categories in ways that some of the scientists I studied called unscientific, sloppy, lazy. So you can see that they had their own conflicts. Those are not my words. Those are their words. So I chose to study the laboratories whose work was respected to find out if they used race and why. Statisticians would say that I have a biased sample. I'm married to a statistician. And we do have a biased sample. But I think that our research adds a dimension that needed to be introduced into the debates. So combined with the research of some of my other colleagues, I think we now have a better picture of who is using race, why, and how. So for this study, I organized an interdisciplinary group of researchers. Ramya Rajagopalan got her degree in genetics and is becoming a sociologist of science. Shel Dotson is a statistician, and Pilar Osorio is a law professor in Madison, and she's also a biomedicist. So this is Pilar on the right, Ramya on the left, and Shel on the bottom. And we've studied, as Lisa said, 5 different sites where research is conducted on genomics and disease, or population genetics. And each of these sites is part, I want to make note that we're not talking about little laboratories, we're talking about groups that are part of one or more consortia. So they're part of enormous networks. Okay, now why have notions of race, ethnicity, and ancestry become associated with genetics and genomics in the last decade? Okay, I already did my turn. Okay. So as the Human Genome Project was nearing its official end in the 1990s and early 2000s, a number of genetic projects concerned with the biological basis of human population differences began to emerge because they believed that these differences could be used to study genetic disease causation. Researchers in the field of molecular biology, genetics, genomics, statistics, and computational biology were developing new technologies and methods for looking at genetic differences between individuals and between populations. So the focus on difference, of course, surprised many non-geneticists, in part because human genome researchers had estimated and emphasized a high degree of similarity between human genomes. And so that's what was called the Human Genome Project. The effort to characterize genetic differences between and among populations itself might not have created a storm of debate, since population geneticists have been conducting such studies for some time now. Their primary interest is to investigate human demographic and evolutionary history. The debate arose because populations used in some of these biomedical studies were often represented by sociocultural terms like tribes, races, ethnic groups, and nationalities. So why have biomedical geneticists chosen to study genetic variation among populations? In contrast to population geneticists, biomedical geneticists are interested in genetic bases of human disease and variation in drug response. So I'll focus on this set of scientists today. So these include Francis Collins, who was then at the Human Genome Research Institute and who is now the director of the National Institutes of Health in Washington, D.C., and who funds a lot of biomedical research in the country. Eric Lander is, he started out as a statistical geneticist, and he heads the Broad Institute at, it's in Cambridge. It includes researchers at Harvard and MIT, and researchers at the Department of Energy in the US were also interested. These geneticists believe that identification of genomic variations between populations would be critical to identification of disease risk genetic factors. So remember that I'm talking about risk here, okay? I'm not talking about causation in the rest of the talk. During the mapping and sequencing of the human genome, scientists estimated that there was at least 0.1% difference between human genomes. And researchers at the Craig Venter Institute and their collaborators have since raised that estimate to 0.5%, and I think we're somewhere in between at the moment. Does anybody know whether we've gone beyond 0.5? Okay, that's good. But whether the percentage difference is 0.1 or 0.5, it's a small percentage difference, but researchers argue that it could still potentially translate into biologically significant genetic variability. So, now why did they believe? Why did these geneticists believe that they needed to characterize genetic variation among populations in order to study disease? So in order to explain this, I'm going to discuss one set of studies that use human population genetic variation to search for disease genes. These studies are called GWAS, or Genome-Wide Association Studies. And there are other kinds of methods that we can talk about, and I'll gladly answer questions in the Q&A, but I wanted to focus on GWAS today. So I'm going to, as I wrote here, I'm going to talk about our partial results. There are many results, and I'm going to try to hit some of them at the end, but I want to focus on one result here so that you'll understand why I go into all of these details about these studies. So one of our conclusions is that GWAS researchers believe, GWAS researchers we studied in several sites believe and argue that they do not use race in their genetic analyses of DNA samples. Okay, no race. We are very curious about this group. So instead, these researchers argue that they have developed techniques that allow them to do their research without using race. Their methods incorporate techniques for producing what they call genetic ancestry, which is not race and is not ethnicity, or any other sociocultural term that we social scientists might use. These techniques now have become state-of-the-art in their field, but with complicated downstream effects, and I'll talk about that later. So what is GWAS? GWAS researchers search for genetic markers in genomes that might be causally linked to common complex diseases, such as heart disease, all the different cancers, type 2 diabetes, asthma, all of the diseases that we see in all populations. So GWAS researchers assume that these diseases are likely to involve small combinations of influences, I should say small combinatorial influences from many different genes distributed throughout the genome. So this is not like we can go in and identify one gene. It will involve genes throughout your genome. Does everybody know what a genome is? Yeah, okay. So genome refers to your entire, all of the DNA in your body. So it refers to, so we have 23 chromosomes. It refers to all of the DNA on all of your chromosomes. So it's all of your genetic material. That's a lot. I think it's 3 billion base pairs, 3 billion DNA base pairs. So that's a lot. If you're, well, I'll go on and talk about it, but it is a very complicated thing to study if you're searching for disease causation factors across the genome. So the researchers that we studied also acknowledged that many other non-genetic factors and processes are involved in the cause of common complex diseases. One of the researchers told me that genetics could perhaps explain 5%, maybe hopefully 10% of a causality of complex diseases. But because they're geneticists, their focus is on the genetic component. So to be clear, I'm not talking about the rare, inherited, single gene diseases like Huntington's, sickle cell anemia, cystic fibrosis, the diseases that have been studied through family linkage studies. Those rare genetic diseases have been relatively easier, I didn't say easy, easier to study because researchers can follow the disease within families. Also, GWAS, Genome-Wide Association Studies, assess the risk for disease. They're not looking at causes. Okay, so keep that in mind as we go. So these researchers assumed that their research might tell them where to search in the enormous genetic components that we have in our body. So where to search in depth in the genome for disease genes. Okay. Until Until recently, common complex diseases have been difficult for geneticists to study because they tend to appear in all populations. That's why they're common complex diseases as compared to the rare inherited diseases. And these diseases are also assumed to involve many genes. So one of the early arguments of the Human Genome Project was that geneticists would be able to walk down the genome and discover, for example, cancer-causing genes. Unfortunately, walking down the genome didn't pan out. Cancer proved to be much more complicated than that. As the human genome was being mapped and sequenced, it became clearer that the common complex diseases were exactly that. They were complex in etiology, and so the language switched from finding genes to finding genetic risk factors, which in the long run would hopefully lead to finding genes. So in order to find disease risk factors, research technocrats began in 1997 to organize a new research design for GWAS. The research design of GWAS began many years before the first experiments happened, and about 10 years before the first results appeared. Because the technologies required to conduct the studies also had to be built, the materials required had to be collected. So the first results of GWAS studies appeared in about 2007, just when I happened to be doing field research. So I felt very lucky. So you can imagine the time and the effort and the money that went into, and I should say the love. People really, put a lot of love into their research. So all of this went into building the tools and collecting the materials needed to do GWAS. So GWAS are large-scale studies that use multiple tools, and especially what they call high-throughput technologies. High-throughput means that they run many samples at the same time through fancy machines. Researchers use new genotyping and statistical techniques to examine large data sets in the search for disease risk genetic markers. They require lots of blood from lots of people, both the healthy and the ill, to conduct their searches. And in fact, I should say that the initial costs required to build GWAS studies involved billions of dollars and raised the ire of some molecular biologists. We're all fighting each other to get money to do our research, so this is normal. Some of the tools needed to conduct a single GWAS include the human genome reference sequence, various databases with human genome sequences and annotations. And here I show the University of California Santa Cruz genome browser. Cheaper genotyping technologies, because at the beginning they were way too expensive. Microarray technologies like DNA chips. On the left is the DNA chip produced by Affymetrix, the Affymetrix company. The human haplotype maps, and as well population genetic techniques. So, as I said, lots of blood is also needed to conduct GWAS. There are so many vampire shows on television, so I'm emphasizing blood here. I think people can relate to. Blood collection is a story in and of itself, and I'm not going to go into the details of it, but I'll gladly answer questions. Now, once the blood is collected, it's then processed and genotyped. So I want to show you some of the. So these are some of the tools used. The left again is Affymetrix, and this is the Affymetrix scanner that reads it. This is another more Affymetrix readers. This is the Illumina chip, which actually has been reading out the Affymetrix recently, and this is the Illumina reader. So, a genotyping center will just have tons of these machines in a very, very large room. So it's a very different kind of laboratory than an experimental molecular biology laboratory. Okay, so after they collect, process, and genotype about blood, then GWAS researchers statistically analyze differences in the frequency of genetic markers between cases, so that's the people with the disease, and controls, those who don't have the disease. Since this method is based on statistical associations, the power that comes from having high numbers of samples is critical. Researchers genotype and evaluate hundreds of thousands of genetic markers per individual in thousands of individuals for association with disease. The numbers vary, but generally they need many to gain statistical power, and 3,000 is the largest we've seen. So 1,500 cases, people with the disease, and 1,500 controls, people without the disease. Okay, since the main focus of this talk is how the researchers managed to get the work done without using race categories, I'll focus on those particular tools. The researcher, I wanted you to get a sense of everything that's used in this research. So the researchers argue that some kind of adjustment for population is necessary to ensure that they're actually measuring differences related to diseases as opposed to other kinds of genetic differences that may be non-randomly concentrated in their cases versus their controls. How many of you like statistics? Oh, cool, they're quite some. Okay. For the rest, talk to them at the end of the talk. So in other words, to minimize false positive associations, the researchers need to account for genetic differences between cases and controls that have nothing to do with the disease. And that's all you really need to focus on. So they call this adjusting for population substructure, or population stratification. You know, we academics like to make fancy names for what we do, and that's what it is. So another way to put this, in their words, is that it allowed them to genetically match cases and controls. So given that common complex diseases might involve many genes throughout the genome, the researchers needed to search throughout the genome. And given a search throughout the genome, they needed to account for other kinds of genetic differences that had nothing to do with disease. So this is why they needed to deal with populations. So, okay. So keep that in mind. That's the main reason why they were dealing with groups in the first place. Okay. So this is the term adjusting for population substructure, genetically matching cases and controls. So to make these adjustments, a group that included a mathematician, a population geneticist, and a medical geneticist developed a statistical software program called Agonist. Agonist uses a mathematical algorithm called principal component analysis and generates genetic marker variation scores for each individual to adjust for population stratification. So this tool development was an intentional effort to avoid using race categories. The medical geneticists I interviewed were enthusiastic about Agonist, the Agonist program as their tool of choice for two reasons. First, they believed that it was a better measure, or as they said, it was better science. Second, it allowed them to not use sociocultural categories like race or nationality. And this technology erases the need for constructing, delineating, and labeling populations that's part of previous study designs. And yet it still allowed them to adjust for population stratification. So everybody was very excited about this tool. But despite the medical geneticist's insistence that they did not use population categories, in other words, groups, including race in their practices, some of them emphasize that their program affords them a genetic standpoint from which to infer population history. Some said that they believe that the program allows them to let the data tell them the genetic history of the individuals whose variation scores cluster together. And I'll show you some of those clusters. So some invoke the concept of ancestry And we're very comfortable using terms like genetic history and genetic ancestry. But like population, ancestry is a loose term that has different interpretations and meanings in different contexts. Eigenstruct outcomes don't necessarily mean shared ancestry. People could have similar eigenstruct spores that come from distant geographic locations without any shared ancestors. So I want to be clear here that eigenstruct spores do not depend on pre-labeling or pre-sorting samples by race, ethnicity, nationality, any of that. But the population geneticists included in their software package an algorithm that allows them to cluster individuals into similarity groups, genetic similarity groups. We put the name genetic variation clusters on it, but it doesn't, basically it's genetic similarity groups on the basis of their pattern of variation in genetic markers located throughout the genome. So I want to show you some clusters. So this is on the right is a set of clusters on that are made from samples taken from people in Japan. Okay, so you'll see that the red is Hondo, which is the main island of Japan. I did this before the earthquake, so I just will note that. The green is Ryukyu, which is the Okinawan part of the country, which of course has always been, there's a history of Okinawa being separate from Japan and then being conquered by Japan. And there are still issues about whether they should be there on the American bases in Okinawa. There's all kinds of problems. Let's see, the yellow at the top is so that those are Chinese samples. Okay, well I'll stop there. So this one did not incorporate Korean samples at that time. So okay. So are we going to live stream during the question and answer too? But we can decide about that. And then I'll tell you more stories. Let's see. The population geneticists, well, I should say that the researchers we study differed on how they used and interpreted genetic variation clusters. The population geneticists felt comfortable attributing geographic meaning to differences in genetic marker frequencies, and they interpret these geographic differences to mean different ancestries. They made the conceptual leap from clusters with shared SNP patterns to infer relatedness and relationships. So, in other words, they assume that individuals with similar variation patterns, genetic variation patterns, as constructed by their tool, also share ancestry. The population geneticists at this site were willing to publish their cluster plots and even assign a group label usually based on nations, and sometimes based on areas of continents to these plots. They were willing to well, I won't go into that. But, so I'm not showing you their clusters. I could show you their clusters, but I wanted to use this one because I'm going to use these to make a point at the end. But the using the eigenstruct program does not require that you actually label clusters. And it also does not require that you publish clusters. Medical geneticists, as versus the population geneticists, argued against using the cluster plots in their articles, in their publications, They thought it was fine to use clusters as a quick check of their samples and to make sure that the genetic methods they used and the computer-generated scores were not totally off base, but they refused to publish the clusters. They argued that with the population geneticists about publishing clusters, and they settled the disagreement by having the population geneticists publish their methodological article separately and in another journal. And they used the population geneticists to publish their cluster plots in that article. The medical geneticists refused to include cluster plots, and they published their key article on their findings of genetic markers related to disease in a separate article in the journal Science, and they did not include the population geneticists among the authors. So they were trying to separate themselves from the population geneticists who published the clusters. So some of the medical geneticists interpreted the plots as clusters of sample that share similar patterns of genetic variation as constructed by their tool, but they did not see this as necessarily shared ancestry. These medical geneticists said that their GWAS research did not use race or ethnic categories in their genetic analysis, nor did they need to, and that the Agonist program automatically adjusted the population substructure. They said there was no need to determine or mention specific ancestries or geographic origins when using Agonist and GWAS. Now that I've shared all these exciting details about GWAS with you, I want to end with some general conclusions from our research. So to repeat, I don't know whether to call it a conclusion or a discussion point, but I want to talk here about the diversity among the geneticists. One of our findings is that there are attempts among geneticists to not use race categories. These researchers view race categories or even ancestry categories as incorrect methods for searching for disease-associated genetic markers. They agree with bioethicists and social scientists who argue against the use of race in biomedical genetics. But again, I want to emphasize the diversity among geneticists. These particular biomedical geneticists don't use race, but others do. So I don't want to lead you to think that the researchers I just talked about represent the majority of geneticists. In fact, we don't know. At this point, no one has done a survey of all geneticists to see who does and who doesn't use race categories. And that's a very difficult survey to do because people have very different meanings of race. But for example, I want to say that a generation of admixture mapping geneticists do explicitly use race categories in research for disease genes. And I'm writing about that in the book. Admixture mapping admixture mapping uses race categories in their researchers who use race categories in their analysis of genetic variation for association to diseases. And they believe that certain racial or ethnic groups have higher incidence of certain disease, especially prostate cancer and asthma, which may be partially explained by the ancestry or the genetics. So, for example, Esteban Richard does research on asthma, and an anthropologist named Wayne Wiley studies Richard's research on asthma. A lot of researchers do study prostate cancer among African American men because African American men have high incidence of prostate cancer, and they use admixture mapping to study it. And so admixture mapping argues that particular groups, especially Latinos and African Americans, have different kinds of genes in their genomes, and that they can actually look for disease-causing genes by looking at the differences amongst those genes. But some GWAS researchers have argued that GWAS technologies will replace admixture studies, that admixture studies are a thing of the past, and that GWAS actually does the search for disease genes much better than admixture. In addition, population geneticists are also committed to constructing populations, and most admixture mapping projects involve population geneticists. But here again, there's diversity. Some population geneticists we've interviewed don't believe that race categories are useful in genetics research, even though they believe in genetic ancestry. So, and I want to remind you that these are all credible geneticists with good reputations in the field. So there is diversity. So I want to move on to conclusion or discussion point 2, the focus on difference versus the focus on similarity. So the cluster plots I've just shown you are based on genetic variation differences at points in a person's genome. These plots make differences stand out. Okay? So one could then start to ask, Joe, why don't you see that there are real differences based on these plots? So that's why I need to show you figure 1. There it is. It doesn't always show up when it's posted. So figure 1 shows 2 random individuals differing at 2 genetic markers, or SNPs for the geneticists there, in about 2,200 DNA base pairs. So the 2 red spots are points where there are differences in the genome. Well, actually, this is chromosome 7, so on chromosome 7. So I want to remind us that the total number of such spots of difference, or hotspots of difference, in the genome make up most likely 0.1% or 0.5% of our genome. And that if, in that 0.1% or 0.5% of the genome, most of the genetic variation markers have no effect on phenotype. So that means that we don't see any effect of them in our makeup. So if one made a cluster plot of the other 99.9% or 99.5% of sample genomes, there would just be one cluster. So as opposed to the clusters that I showed you earlier on the right, if we just focused on the 99.9%, you would have one cluster. You would not have these different groups. So the clusters are based on points of difference. And there are very few points of difference. Okay. Now, let's see, where am I? The reason that the medical geneticists didn't want to publish their plots is because they see it just as a tool in the process, and that if they published it, this tool could be misread to emphasize differences between groups, since it focuses on difference. The population geneticists, on the other hand, focus on difference. That is their job. They construct populations. And so they're perfectly comfortable with emphasizing difference. So it's part of the different lines of work, the different occupations of academics or academic researchers that lead them to different kinds of commitments. Okay, conclusion 3. I argue that this invention of technologies that can be used in place of race categories has also created a social space for making choices not to use race in contemporary biomedical genomics. Through their discussions with social scientists and because of their personal commitments, these geneticists, the medical geneticists we study, are facilitating a potential transformation of genetics research. In other words, the availability of this technology puts the onus, or should put the onus, on biomedical geneticists to not use race when analyzing their data. Because they have this tool, they don't need to use race categories. So one could argue then that a byproduct or an unintended consequence of GWAS studies is the invention of a way of accounting for population differences without using race. In other words, GWAS were intended to find genetic markers associated with common complex disease, but its practitioners also developed methods for producing genetically similar populations without labels. And it's unclear what these researchers and their readers will make of these genetically similar populations. Humans have managed to stratify societies using all kinds of difference. But one potentially positive outcome is that biomedical geneticists can do away with using race or ethnic categories in their work. Okay, now I want to move on to conclusion 4. So here I talk. But another finding of our research is that there's an intertwined relationship between the two concepts of race and ancestry in 21st century biomedical genetic research that makes it difficult to escape histories of socio-political constructions of difference. So for example, although some of the GWAS scientists I studied attempted to avoid using race categories in their work, their technologies and practices invoke concepts of genetic history and ancestry that are difficult to disentangle from race categories. Genetic history and ancestry are concepts based on complex and contingent practices and technologies of assessing population differences, but nevertheless are oftentimes read as synonymous with notions of race by some audiences, by my mother. So it's not, it's just, it's something that we do. So Francis Collins, too, the former director of NHGRI and now the director of NIH, is himself cagey about the distinction between ancestry and race. In 2004, and I think I haven't asked him in 2011, but in 2004 he published a very careful middle ground, albeit by arguing that because, quote, ancestral origins in many cases have a correlation, albeit often imprecise, with self-identified race or ethnicity, it's not strictly true that race or ethnicity has no biological connection. So he said this in 2004. But I also want to note that Collins uses the term biological and not genetic. The two are not synonymous. Biology and genetics are not the same. The problem may lie with population genetics. The population geneticists easily move from SNP variation cluster to genetic history to shared ancestry. And while the first term is based on genetic marker similarities, the other terms imply belonging and relatedness. Some population geneticists interpreted these results to mean that individuals within a cluster share more than just genetic patterns, that they actually have shared ancestry. This belief is based on theories about human genetic human migration patterns in their field of study, which themselves are constructed through histories written by archaeology, physical anthropology, linguistic anthropology, and social anthropology. So we use this term, genome geography, to refer to the practices of linking genetic variation clusters with migration patterns across continents to in turn define ancestry. So in other words, in addition to genomic technologies, genome geography is constructed from histories of scientific and sociocultural discourses on geography, history, and migration. Again, this was before the quake. I'm very upset and sad about what's going on in Japan. I have relatives there and friends there. But, so I may look like I'm picking on the Japanese, but it was really good work. So, for example, a study conducted in Japan used the Agonist software to distinguish different populations among Japanese citizens, including those who were labeled Japanese, Okinawan, Han Chinese, and now Korean, but I don't have the Koreans on this slide. Note that these are national and ethnic categories. These are not, well, okay, so just leave it at that. They're national and ethnic categories. The study concluded that the design of future GWAS must take into account the genetic differences within the Japanese population. This is an interesting conclusion and rationale for conducting the study, especially given that many geneticists outside of Japan regard and treat Japanese samples as coming from a roughly homogeneous population. The study also confirms already present underground and sometimes overground stories about national and ethnic differences in a culture stratified by ethnic differences and prejudices. For another example, against earlier Scandinavian histories and linguistic anthropologies, which designated Finns as non-Indo-Europeans and distinguished them from Scandinavians, the Danes, the Norwegians, and the Swedes, a Finnish study used population genetics to conclude that Finns are really more closely related to Europeans and are not descendants of Siberians who were thought to be of Asian origins. So here, we have these stories changing, stories of human migration patterns and origins changing with DNA. So my point is that the presumed stability of DNA stabilizes and destabilizes cultural stories and social histories. But these genetic stories through genome geography are also already constructed using cultural assumptions and social histories. So for my last conclusion, I'm moving back to disease. I want to return, this is a very dangerous set of statements I'm going to make, so I'm quite willing to discuss. I want to return to the question of what we can say about the genetics of these disease causation. So first I want to make clear that when we talk about common complex diseases, genetic evidence thus far has helped to confirm that we still know very little about genetic causes. GWAS studies are correlational studies that have provided some evidence that the presence of certain genetic marker variations slightly increases the risk of certain common complex diseases. So that some are cancers, some are diabetes, type 2 diabetes. So GWAS is all about risk, not causality. Molecular biological study of the functions of the genes where specific genetic markers have been found may tell us more in the near future. Some of the other significant markers identified by these studies are not located in genes. It's unclear whether they're active agents, they're part of active agents, are near active agents, or something else altogether. So given new technologies and more information, the role of genetics in disease causation has become even more complicated than ever. Researchers are now beginning to look at rare variants and copy number variations and more. On other fronts, systems biology research approaches have begun to grow. These approaches look at bodies as complex systems of interactions among many kinds of environmental, genetic, and biological factors. Socioeconomic factors are sometimes considered to be synonymous with environmental factors. Systems biologists predict that their findings will reveal far more effective ways to treat and cure human disease and to stop or slow epidemics of infectious disease. So in short, I'm arguing that the understandings of a genetic disease and the genetics of causality have changed dramatically with recent genomics. While there are more questions than answers, it's clear that DNA and genes have come to play what I'm saying, and this is where it's going to be controversial, is a smaller role than previously thought in disease causation, at least for common complex diseases. And so second, and related to that, remember that Francis Collins said that it's not strictly true that race or ethnicity has no biological connection. Collins uses the term biological versus genetic. The two are not synonymous, as I said, and that's an important slippage he's making. In other words, as many epidemiological studies have shown, social epidemiological studies have shown, there are racial disparities in health outcomes. Here I specifically refer to social categories of race. But most social epidemiologists argue that these disparities are due to other kinds of disparities. In income level, education level, levels of stress, perhaps due to racism, access to health care, access to good food, fresh produce, access to exercise, and all that. So all of these socioeconomic disparities probably have much more power to explain biological differences or health outcome differences between social race groups than the genes. And so thus race may play a role in disease causation, but not because of genetic differences. Third, this difference between genetics and biology is crucial for policy analysts and policy makers to understand. Efforts to treat disease and improve health outcomes need to focus on social, economic, and political causes and not necessarily on genetics. And I'm not the first person to say this. I'm raising this as a point. Fourth, some of the geneticists we've chosen to study understand this, and they do acknowledge that the cause of common complex diseases, the causes are primarily not genetic, but they're geneticists, and this is what they have to contribute to disease research. There has been much outcry about the failure of GWAS studies to find causal factors, at least in the US. But these researchers told me that their research might, again, be able to explain perhaps 5% of risk, and that that's a contribution. Okay. But, you know, it makes clear that they assumed that it would not contribute that much, but they were hoping. They really were hoping. Okay, summary, take-home points. First, I provided some evidence to show that the revival and reification of race in genetics is uneven. That some geneticists clearly believe that the use of race in their research leads to incorrect science. And that they've developed tools to make it possible for them to do their research without using race or ethnic categories. Second, unfortunately, I also show that the three concepts, race, ancestry, and genetic ancestry, oftentimes become confounded with each other in minds, languages, and policies. And we call this confounding genome geography. I want to make clear that genetic ancestry is a statistical product that should be distinguished from social concepts of race and ancestry. Third, I've provided brief examples of how new genetic technologies are not always used or interpreted in the same way that their designers intended. Some downstream users of Agonist are using it to revise older stories of human origins and ancestral lines and to transform our ideas of who our near relatives are. And so we coined this term genescapes. These researchers, so the Japan cluster plots I showed you were designed by these Japanese researchers who used Eigenstrat, but they thought that so they used Eigenstrat to actually create clusters and publish their clusters, and they thought it was very important that people understand that there are differences within the Japanese population. So just as landscapers design new landscapes, geneticists are redesigning our origin stories and our relationships with others. Moreover, these new stories and relationships are presented as the result of genetic analyses, but they also include stories of race, ethnicity, migration, nation, origin, and all that. Fourth, I argue that we all need to revise our understandings of the genetics of at least common complex diseases to give it a much smaller place of explanation and to return to socioeconomic explanations of disease and health disparities. And finally, I hope that our analysis may provide and promote theoretical, pragmatic, and constructive ideas and practices for helping at least social scientists and others join in the discussion of race in the age of genomics. Thank you.
things are. But now, first, we have to learn it. So please.
There's a microphone right there.
Well, I was controversial, so some of my colleagues disagree with me about, especially the claim about disease.
It's interesting, but I'm kind of wondering whether you know the words of Augustin Fuentes in Notre Dame. He's an anthropologist who has been sort of thinking about, I guess, epistemologies. And so what you're talking about in terms of the sort of dynamic relational status of becoming in the world, and I kind of felt like you weren't getting there all the way. In that you're kind of missing out on having epigenetics as a possibility of sort of addressing human rights and kind of taking it along an avenue that will kind of open it up to an engagement at a much more political level.
Can you say more about, because epigenetics is, we're actually studying epigenetics now. Epigenetics can mean studying the histones around the DNA, or it can mean studying.
I guess I'm kind of blowing it up beyond the relational. So where you tagged me was the sort of the last line in your previous slide around the difference between biology and genetics. So the tip of say my adopted daughter, who may not have any of my DNA, really has my biology because she's growing up with me, and she has my kind of relations in the world, and has taken on my biology. And I kind of wanted to hear you kind of expand on that. At least that's my understanding of the difference between biology and genetics.
Well, yeah, I mean, biology is enormous. So, for example, I was just showing a group of people at dinner these slides made by someone who calls himself, what does he call himself? I guess he called himself a systems biologist, and he studies the gut microbes, so the microbes in the stomach. And he showed a slide where he made clusters, but the clusters were based on the bacteria in the stomach, and he had Japanese, Chinese, and American. And so when I first saw that, I said, "What is this?" And basically, he was showing that the bacteria are different because we eat different things in the U.S., Japan, and China. But that was just one sample, one set of samples he had. So that's one way of constructing clusters or groups of people who are related to each other on the basis of what we eat. Right? So that's one kind of biology. He calls it metabolomics. But I think you're talking about something else. So I mean, obviously the example that I was giving with respect to a lot of poor communities in the U.S. is that you will see racially segregated disparities. You see a lot of poor health outcomes among African Americans, in part because they're just not, their diet is adequate, they're not getting good health care. And one of my colleagues actually argues that this is in part education, but also in part income. And so their biologies, their physical health is not good not because of genetics, but because of their general everything I need: exercise, diet, nutrition, access to health care. So those are all of those things make differences in our biologies.
So I guess where I'm getting at is that genetics, but it seems that we've gone past genetics now, and I'm kind of curious why everybody's still focusing on genetics, and I'm going to throw that into so we've gone into proteomics, we've gone into epigenetics, and these are explaining so much more in the ways that gain a kind of a platform that accepts in issues that we can deal with politically, like human rights issues and class differences and all of these other things. And I guess I'm just wondering, why stay on genetics?
Oh, I still love genetics. I think it's really interesting. So it's not like, so I'm not saying that, okay, so this is a workshop announcement that I got that says conquering the complexity of protein data. Genes are easy. And so then I wanted to say, no, not. Genes are not easy. So first of all, despite what I'm saying, I'm not saying that we shouldn't do genetic research. I think that genetic research is interesting. I agree that the world has gone on. We're RNAi. My postdoc actually got a PhD in RNAi, and there are so many different RNAs. The proteins are really complex. We're studying systems biology and epigenetics now. So yes, I mean we're moving beyond, but a lot of all of this research is actually based on genomic research. So they are using all of the genome information that was produced through the Human Genome Project, the HAPNet project, and all of that. So it's not as if they're just kind of ignoring genetics. Genetics is still part of it, and is used in the other research. I'm just arguing that when we're looking at common complex diseases, if, say you're an economist, I'm at the Russell Sage Foundation, and there are 6 other economists, 6 economists among the 19 of us there, and they're always talking about, well, I shouldn't put words in their mouths, especially when we're live streaming, but what do you get for your money, right? And so are we really going to get the kinds of solutions to our problems around, trying to treat and get rid of common complex diseases by investing more in genetics research, or are you going to get it by trying to improve the education and health? I mean, education of and access to all the things that you need to be healthy. So that's more with respect to common complex diseases, I would say. If you're talking about specific rare genetic diseases, it's still very important to do genetics. I would not say don't do the genetics for that. So I think we just need to be aware of what we're investing where. I guess that's what I would say. Okay. Yes.
Just following from that, I was wondering how much what live streaming that you showed. Do you mind coming up with that?
And now experiencing physiological difficulties. So I was wondering about, Lauren, what you were saying about the what's driving the research. I mean, what you're saying is we really know what causes these common complex diseases. We know that poverty causes them. We know that a bunch of environmental factors. We know certain lifestyle issues and choices. We know what causes these diseases, but we don't want that answer. What we want is an answer that's going to help us drive scientific innovation. So let's see what new technology we can build. We want something that's going to, we want the type of answers that are going to push forward the types of new science and new technologies that are sort of part of the machine. I mean, is that the sort of take-home message here, that we're going to move on to proteomics because we're going to have a whole new bunch of fancy machines, and there's going to be a whole bunch of new science, and we'll find that proteomics can tell us what 3% of the causation is. So we know all the environmental, social factors. It's like 80% of the causation. We've got 5% genes, 3% proteins, and you're just going to keep on adding what are, useless for any kind of medical or social policy. So as a sociologist of science, is that the message I should be taking home from what you're saying?
Yeah, I'm very nervous about this message. So first of all, I want to say that when he said 5%, he really wasn't talking, it was a cartoon, right? Because of course everything is very, very complex. And so he didn't really just exact 5%. But I'm kind of caught here because if I were designing policy, then I would say yes, put all your money into improving education, access to health care, access to... And there's some communities that don't have stores that sell fresh produce nearby in the U.S. And so, I would put all of my money there. If it were my money, I would put all of my money there. On the other hand, I'm also very fascinated by science, and I find it really interesting, and I like my job studying science. So I'm kind of caught here. So I would say that with respect to common complex diseases, I would say yes, if I were God, if I were God, anyway. So, and if I were, if I could, gain control of the policy making in the U.S., I would certainly put all of my money, not in genetics.
But aren't you just then saying that basic science is cool, and we get to find out cool stuff? And I'm all over that. I love basic science, I have no problem with that. But then the justification for doing what is, in effect, basic science is this big social policy lie that, oh, we're going to learn how to cure diabetes, or we'll figure out the genetic basis of. And so then it sounds like what you're saying is, well, yeah, we've got this big lie going on, but don't worry, because science is cool, and we're not going to get this done otherwise.
Well, I think that they actually hoped that they would find more. I mean, they really did hope that they would find genes for common complex diseases, and that they could use that, if not, to actually produce therapies, because of course that's a difficult job, to then be able to, say, look, you have this gene, and so you better clean up your act because you really are susceptible. Even if it's 5% susceptibility, which of course is a very weird way to talk about it. But so I don't know. I mean, did they convince themselves that they really were hoping to find genes to be able to fix things? I don't know. I didn't ask them that. That's a good question. I'll have to go back and ask that question. But yeah, the science is cool. I don't know, maybe we should open this question out to the scientists in the room. I mean, what do you think about this issue? You're going to be on the spot very soon.
Okay, only being in my second year of Bachelor of Biology and the History of Science and Technology, I'm certainly not a scientist, but I kind of think that you're giving science a bad rap because we don't, by any means, know exactly what every codon codes for. We don't know what every protein that is made by the genome in every human being does, and we need to. And I think that the more we study genomes, the better we'll come to know a bunch of pathways that even if it's not okay, you have this gene, so you're going to have this disease, you're certainly going to gain a deeper understanding of how the complex pathways that affect complex diseases work. So even if sequencing genomes and learning that only 5 or 10% of genes really affect diseases still means that you'll have kind of a concrete, not checklist, but okay, this is what, imagine a world where we knew what every gene did to the body, and then you could look at a person with a certain genome and say, okay, so based on your genetics, this is what kind of should be happening with your body. And then you could take that knowledge and add your knowledge about the person's socioeconomic status or lifestyle. And then that would, in my opinion, be the ideal situation. So I don't think by any means you should talk of science is just cool and not actually helping the situation. Because I think although you want to take one thing into consideration, you also want to have the kind of concrete knowledge of what a genome does. And then with both of those things, you can work together towards a better system of health for everyone. But I think you need both things.
Well, I actually, I mean, absolutely, the systems biologists, the people who are looking at pathway analyses, they use genes, they use proteins, they use RNAs. They're actually looking at these complex metabolic pathways and disease causal pathways. So, and they're just beginning. I mean, it's really not. They really are just beginning, and they did need a lot of genomic information to do this. But I think that this will help us to understand. If I were a policymaker, and if I wanted to fix health outcomes now, or very soon, I would start with investing in education and all the other things that we talked about. And because as much as all of this understanding is really interesting and valuable, it's going to help us understand that for now, but it's not, any kind of actual interruption or treatment of these pathways is going to take a long, long time to get there. Unless we're lucky, unless they're lucky. That's another possibility. I mean, a lot of our therapies came from luck, right? More than understanding the whole causal pathway. So, but that's my opinion. This is just my opinion on this. So I value your opinion. You're the future, so.
I'm just wondering, in your study, like in terms of studying in the lab, did you find that anyone was concerned that looking for genetic causes for these common complex diseases, that there could be like malicious uses of that? So for example, I would think the first thing that would come to mind for me is things like insurance companies, as genetic testing becomes more and more common, insurance companies using genetic testing and risk factors to assess different like insurance premiums or to refuse to insure someone, or other similar, like some of the resistance to publishing data about how you can divide people or how you can cluster people, concerned also with the ways that those factors could be used maliciously?
No, I mean, none of them were, nobody talked about that. And in part because we're not there and we don't really, we have SNP markers. In fact, it's probably gone the other way. A lot of the private companies that have developed in the US have taken some of this information produced by these studies to market them, to sell tests to regular humans like me to tell me whether I'm susceptible for this or that, and to get clean up my act, so that I don't develop certain diseases. There are many problems with these companies. First of all, we don't know what the SNPs mean, and also and also they're very their data is based on proprietary databases, and there's just a lot of problems with this. So but what's funny is that nobody really was concerned about what you're talking about there. We're actually writing articles concerned about what people are going to spend all their money on these these tests for SNP markers or risk factors, and that it's a waste of their money. So now there is concern that it's possible to identify people by their genetics, and that a new technology was developed very recently in the US that actually will be able to identify people if their genetic information is put on display in websites. So the researchers originally put all of their data, because they're funded by NIH, they put all of their data on websites. When this technology came out, they had to basically not privatize, but they basically had to put a protection on their database to allow only scientists, reputable scientists, to access the data because it was possible to identify who the person was based on that genetic information. So it's a great problem. My colleague Clara Sorio is writing a paper for a law review article on whether it's possible to have anonymity anymore in genetics research. So yeah, so I mean, certainly for the U.S., it's a really big problem, and it's a big. But the problem of identifying your SNP markers, but it's not enough because you would gain much more information by finding out where you live, what your income level and all that is than you would from the genetics at this point for common complex diseases. So thank you.
Hi there. As a scientist and one of the people that raised their hand that they love statistics, I have to express a little bit of skepticism with regards to the decision to discount genetic basis of the disease on the basis of a 5% accounting variability. I have certainly seen studies of social determinants of health, and those tend to have large variances accounted for in terms of health outcomes. However, when you're studying or when you're deciding between investing resources in a genetics research route or a social research route, you have to look at those studies and realize that you need to, in order to assess the relative importance of those two, you have to put them in a combined context. That is, that 5% is saying, ignoring everything else, including social influences, this is what our simplistic model of genetics accounts for. It may be that if we have more nuanced models of genetics, looking at the proteome, looking at more nuanced interaction between genes, not simply presence absence, that we grow that percent that the genes account for. Additionally, all the research where we have societal, cultural impacts on health, certainly it's the case, or it could be argued that there might be a correlation between the social markers that are used in those studies and the genetic attributes of those individuals. So there might be a proportion of the proportion explained by social factors that is actually the part that's explained by genetic factors. And we need more research in order to really have a good detailed understanding of how those variances partition out.
Okay, thank you. So I will be sending you a draft paper to read so that you can comment on it, because I think that that's how the scientists think, the scientists we study think of it. That in fact, this is not additive, right? It's not an additive process. It's what's the other word? It's not additive.
Interactive.
Yes. And they're looking at, well, they're not particularly looking at it, but systems biologists are looking at the interaction amongst genes and proteins and RNAs and all these. The microbiome project is coming in so that we can look at how bacteria interacts as well. So there's a lot of excitement, and I agree that that's why I said 5% is a cartoon, right? And so I agree with that.
5% is the lower bound. It could be much higher if we get more nuanced models to account for.
Okay, thank you.
Hi. Hello. Okay. I have a question from online. This is from Joan Fujimura. I hope I'm pronouncing that all right. From Seattle University of Washington. So bear with me. I'm asking on behalf of this person. Does the whole genome sequencing obviate the need for ancestry estimation or even PCA-based approaches to identifying disease variants or loci?
I'm sorry. Can I read it? Is it okay if I read it?
Yeah, if you can read my handwriting.
I'd like to hear it again.
Does the whole genome sequencing obviate the need for ancestry estimation or even PCA-based approaches to identifying disease variants or loci?
I'm sorry. So So he or she means having the entire genome, so with the whole genome sequencing.
I believe so.
Yeah. Okay. I think I understand. So I actually asked a bioinformatician who's working on this, and he said they will still need to understand population differences, even with whole genomes. They will have to understand population differences in order to search for disease genes. So that's the answer that I got from some of the people I had talked to. Yes.
That may have to be our last question. I just realized we are completely out of time, but we were too caught up in the discussion.
Very, very good question. This has obviously been mainly about common complex diseases, and a couple of the slides said related to the need to continue to study what we might call high penetrance, non-complex disease. And I fully agree with that. That seems kind of axiomatic. But do you really need to study race with those ones or not? Because that's the original question.
Oh, no, because you're using, there's so many other tools, family member studies, and I think other tools since then, so I don't think race is an issue.
Even with the high penetrance?
I don't think so.
Yeah.
Now, again, maybe the geneticists in the room would like to type up here, but I don't think so. Okay, thank you.
Please help me thank cordially our questions, but we can continue informally and have some healthy food and healthy drink to improve those patterns.